Hi, everyone. In this final section of the cells lecture, we're going to talk about transport across cell membranes. And this is so important, and it's referring to how things get in and out of cells, like nutrients, how does waste get removed? And this is so important because cell transport is really at the heart of everything from nutrient absorption in our digestive system to how our kidneys work, to how to grow bacteria in lab, even to cancer. For example, in cancer, abnormal nutrient transport can support uncontrolled growth. So even though we're still talking about the foundation fundamental level, it's important across so many areas of health. So at the end of this section, you'll be able to understand basically active and passive transport of substances across cells. So this includes things like simple diffusion, facilitated diffusion, osmosis, which are passive transport, and then active transport in cells, which requires energy. We'll understand the impact of different environments on cells. Specifically, we're going to talk about what happens in cells in things like a hypertonic, hypotonic, or isotonic environment. And then we're going to talk about in detail simple diffusion, facilitated diffusion, osmosis, and active transport. Okay, starting off with the plasma membrane. So as a reminder, all cells have a plasma membrane, and one important function of the plasma membrane is to allow things to move in and out of cells. So keep in mind that it goes both ways. A lot of times students think it's just a movement of things in cells, but things go both ways. And we say that the cell membrane is selectively permeable. That means that it selects what goes in and out, and not everything is permeable, meaning it can go in or out. So this regulation is very important. There's two types of transport. Passive transport is called passive because it doesn't require any energy. And the reason why there is no energy required is it's because substances are moving from where there's a lot of them to where there's a little of them. So anytime things in chemistry go from high to low concentration, that's a passive process, meaning there's no energy required. There's also active transport. The word active is there because it requires energy. In biology, whenever I say it requires energy, it means that you require ATP molecules because ATP is energy. And the reason you need energy is because substances are going from where there's a little bit of them to where there's a lot, and that requires energy. So when things go from low to high concentration, that requires energy. That's the same thing as saying things moving against their concentration gradient. That's an active process. Going from high to low concentration does not require energy, and it is a passive process. Okay, so now we will focus on these two processes. There are examples of passive processes and active processes, and we're going to go through each. So with passive transport, there's three types of passive transport in a cell. There's simple diffusion, facilitated diffusion, and osmosis. And we're going to learn about those in detail. And then for active processes, which require energy, there is just what we call active transport. And then there's endocytosis and exocytosis. So before we get into the details of this, I just want to say that prokaryotes and eukaryotes, all cells have passive processes, means they have simple diffusion, facilitated diffusion, and osmosis. And all cells also have active transport. But in addition to these, eukaryotes also do endocytosis and exocytosis. Prokaryotes don't. Eukaryotes do all of these, plus these two. Prokaryotes, meaning bacteria, only do the things that aren't in yellow. Okay, so this is a video before we get into the details of each because I think it sets a really nice tone for understanding this.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
And again, exocytosis
Now
and
let's move to the other
endocytosis
side of our intestinal cell.
are eukaryotic cells only.
Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so that is a very complex video explaining everything that I'm going to explain in detail in this, but I just wanted a video that puts it all together. So we're going to break it down right now. So I'm going to start with passive transport. Okay, all cell membranes, all cells do something called passive transport. Passive transport means that things go in and out of a cell and there's no energy required. There is no ATP required. That's the same thing. And the reason why there's no energy required is because substances are moving from high to low concentration. There's three types of passive transport. There's simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is a process that allows small molecules, things like oxygen, carbon dioxide, to pass directly through the membrane. So diffusion, which we also call simple diffusion, is when they just pass in and out of the membrane directly and there's no energy required. So for example, in our lungs, oxygen diffuses from the alveoli where its concentration is very high into the blood where its concentration is low. So this is a type of simple diffusion. There's no energy required, there's no protein required, and things are moving from high to low concentration. Next, we have facilitated diffusion. Facilitated diffusion is also a type of passive transport, meaning no energy required. The only difference between facilitated diffusion and simple diffusion is facilitated diffusion needs facilitation, meaning it needs help. So it requires a membrane protein. So larger molecules or ions, so things larger molecules like glucose or ions like sodium can't diffuse directly through the membrane, so they require a protein in the membrane. And so that's an example of facilitated diffusion. They require a transport protein. An example of this is like, for example, insulin facilitates the diffusion of the sugar glucose into cells by increasing the number of glucose transporters in the cell membrane. So when there is a problem with this, where we see in this results in elevated sugar levels. So facilitated diffusion requires a transport protein. Next, we have osmosis. Osmosis is just a fancy word for the movement of water in and out of cells. The movement of water is passive. It doesn't require energy. Water is so important. So it has to be able to get into and out of cells. So the movement of water across membranes from an area of high to low concentration is called osmosis. And this is very important. So it's important throughout our body for maintaining blood pressure, for fluid balance, and also it's important for bacteria as well. So here's a video focusing on transport again.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes.
Okay, so I just want to briefly talk about this image. It's very similar to the other video. So for passive transport, things that require no energy, when small molecules like oxygen are just going in and out from an area of high to low concentration without a membrane protein, that's called simple diffusion. Facilitated diffusion is when we require a membrane protein. And again, things are moving from high to low concentration. And then osmosis is just a movement of water. Water can go into and out of cells either just by simple diffusion or by using a membrane protein. So here is, again, just focusing on passive transport. I know it's a lot, but I really want you to understand this.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Okay, so moving on, osmosis. So, osmosis is how water moves into and out of cells. It's the movement of water across the membrane. It's a passive process, meaning it doesn't require water. It doesn't require energy. It doesn't require ATP. And when we think of how water moves across a membrane, water is so important for cells to get into and out of cells that it can just either diffuse through the membrane, and there's also special proteins inside of our cells called aquaporins that are water channels that allow water molecules to move into and out of cells. Now, the movement of water can either enter cells or exit cells, and it does both. But is it entering cells more or exiting cells more? It depends on the type of environment that cells are in or the type of solution that cells are in. There's three types of solutions or environments. We call them hypotonic, isotonic, and hypertonic, and we will talk more about them. So again, before we get into the solution, again, osmosis is the movement of water. Water can enter or exit cells. It goes both ways. And water will always go from where there is more water to where there is less water. So that's the same thing as saying water will always go from where water is highly concentrated to where water is in low concentration. So this is exactly the same thing as saying water moves to where there is less of it Less of it means there's more salts, and I'll explain that in a second. So taking all that aside, let's think of a cell. So inside of a cell, you have water and you also have solutes. Solutes are things like salts and sugars. To make it easier, I'll just say salts, but solutes are salts or sugars. Just think of a cell, there's solutes in it, and there's also water. And then outside of a cell, there's also water and there's solutes. Water will always move. I want you to remember this forever. Water will always move to where there's more solutes, because if there's more solutes, then there's less water. But taking that aside, to understand this lesson and for everything to be tremendously easier, the one thing you need to memorize is water will always move to where there's more solutes, more salts, more sugars. So if there are more solutes outside of a cell, and I tell you, is water going to enter the cell or water going to leave the cell? Water always goes to where there's more solutes, so water will leave the cell. If there's more solutes, more salts and sugars inside of a cell, then outside water will enter the cell. That will help you understand everything forever. Is just remember, water will always go to where there's more solutes. So, osmotic pressure is this fancy word of cells needing to maintain a certain pressure to be healthy. Think of car tires. If the car tires are too filled up, they have too much in them, it's not good because they'll blow up. And if they're not filled up, it's bad because they'll deflate. You need the perfect pressure of tires on your car or on a bike for it to function properly. That's the same thing with cells. And the thing that controls this pressure is water and solutes. So we want a healthy amount of water in our cells. This is partially why having too much salt is not a good thing in someone's diet. So we'll explain that more. So now we're going to talk about solutions and then put all these things together, how this all makes sense. So in science, we say that there's three types of solutions. An isotonic solution has the equal solute concentration as a cell. It matches the salt and sugar concentration of an isotonic solution matches cells. So that's things like IV in a hospital. It matches what your cells look like. Hypotonic solution, hypo means less, is a solution that has less solutes than a cell. Think of plain water. And hypertonic solution is a solution that has hyper, a high amount of solutes, like sugars and salts in comparison to a cell. So these words refer to the solution, not the cell. So if I tell you we put cells in a hypertonic environment or in a hypertonic solution, hyper means overly. So this is an overly salty solution or an overly sugary solution. If I tell you that we put cells in a hypotonic solution, that could be like plain water, it has less solutes or less sugars than the cell. And then an isotonic solution has an equal amount of solutes and sugars like the cell. So, an important question is asked in science, which is what happens to cells if you put them in each type of these solutions? I'm going to start with the easiest one to understand, which is an isotonic solution. So, let's start here. As a reminder, isotonic solution is a solution that has an equal amount of solutes as the cell. So like if you took a cup of water and you made it match the solute concentration of our cells, the same exact salts and the same exact sugars, that's called an isotonic solution. If you make it and then you put cells in it, we ask, are cells going to enter or leave? Is water going to enter or leave the cells? And I told you the one thing I want you to remember forever is water will always go to where there is more solutes. In an isotonic solution, it has an equal amount of solutes as the cell, so water will enter and leave at the same rate. So that's great. So an isotonic solution, what happens is water will move in and out at the same rate. That is called equilibrium. A lot of times students, when I tell them what happens to cells in an isotonic solution, they tell me nothing. And I tell them if nothing happens, you would die. It's not nothing. It's water is entering and leaving at the same rate. So that's great. Now let's think of a hypertonic solution. Hypertonic, hyper means over. It means too much sugar or salt. So if you have salty water, that's a hypertonic solution. Sugary water, that's a hypertonic solution. And you put cells in it, what would happen to the water? Will the water move in the cells or out of the cells? Remember, I told you to memorize forever. Water will always go to where there's more solutes. Because the solution has more solutes, the water will leave the cell and the cell will eventually shrivel in a hypertonic solution. Finally, in a hypotonic solution, hypo means less solutes than the cell. We can think of like plain water. If you put cells in plain water, there are more solutes inside the cell. Water always moves where there is more solutes. So water will continue to move inside of the cell and the cell will burst. That's why overwatering a plant could kill it. So if you didn't know, if you overwater a plant, it could die. The reason it could die is because water will keep entering the cells and the cells will burst. Hypertonic solution, if you put salt on organisms, too much salt, they could die. And that's because the water from the cells leaves the cells and the cells shrivel. So this is important because if we're trying to grow bacteria in lab, we want to make sure we're using an isotonic solution. If we are putting an IV in someone, IV is not plain water. IV has solutes in it because you want it to match the solute concentration of cells. And then just as a fun fact, you can kill a lot of microbes by adding salt water. So this is a summary of the last slide. I'd like you to know what happens to cells osmotically if you put them in different types of solutions. If you put cells in a hypertonic solution, overly salty solution, the water will leave the cells and the cells will shrivel. If you put cells in an isotonic solution, it means that the solute concentration matches the cell. Water will enter at the same rate, so it's a healthy environment. If you put cells in a hypotonic solution, meaning the solution has less solutes than the cell, the water will enter the cell because the cell has more solutes and the cell will eventually burst because of high pressure. So this is important both for microbiology and also for human cells. So think of water movement with red blood cells. If you put red blood cells in a hypertonic solution or a person has crazy amounts of salt to eat or sugar, I'm talking like unbearable amounts to where it could kill you. That's because in a hypertonic solution, water will leave the cells and the cells will shrivel. Isotonic solution, it will match the red blood cell concentration. So water will enter and leave red blood cells at an equal rate and the cells stay healthy. If you can drink water, you can drink so much water that you die. That's called a hypotonic solution. And that is because if you are drinking crazy amounts of water, this is rare, but it has happened. Everything out of balance is bad. So hypotonic solution, if you drink so much water, the water will eventually start entering the cells and the cells will burst. And that is why it's very important to take into consideration IV concentration. And again, this is also why IV is not just plain water. All IV solute concentration matches cellular concentration. So that's that. That's how it's related. And again, this is osmosis and it's the passive transport of water. Now we're going to briefly talk about active transport. So active transport is a process that requires energy. So far, everything I talked about did not require energy. Energy for biology is ATP. So active transport requires ATP. And the reason why you need energy in active transport is this is when you're trying to move substances from where there's a little of them to where there's a lot. That's the same thing as saying moving things from areas of low concentration to areas of high concentration. That's the same thing as saying moving against the concentration gradient. And a perfect example of this is the sodium potassium pump. This is something that is physiologically in us, and it's important for nerve transmission and muscle contraction. So this requires energy. So there's so many things that require energy. Electrolyte imbalances can disrupt this pump. So anytime you are moving something against its concentration gradient, you require energy and you require a membrane protein.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here's how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so that's active transport. So far we talked about passive transport, simple diffusion, facilitated diffusion, and osmosis. And now we talked about active transport. These are present in all cells, in prokaryotes and eukaryotes. Now I'm going to talk about another additional form of active transport that's only present in eukaryotic cells, like human cells, animal cells, but this is not present in bacteria, not in prokaryotic cells, and they're called endocytosis and exocytosis. These are important processes for transporting large molecules across a plasma membrane, and they require energy. So exocytosis is how cells release large molecules or waste products. So things like hormones, neurotransmitters. So it's removing, exiting large molecules. So, for example, insulin is released from pancreatic cells by exocytosis. Endocytosis is taking in large molecules in the cell. And so examples of endocytosis is something called phagocytosis. That is how our immune cells, our white blood cells, eat up pathogens that cause infections. That's a type of endocytosis. And there's pinocytosis. This allows the cell to take in fluid. It's called cell drinking. So here is a picture of exocytosis and endocytosis. So again, exocytosis has to do with rearranging the cell membrane, and it requires energy to make large molecules exit. Endocytosis is bringing things in. Examples of endocytosis is phagocytosis, where you eat up pathogens, and pinocytosis, where cells take in liquids. And so that is our lecture on membrane transport. We covered passive transport, which doesn't require energy, no ATP, and that's because we're moving things from high to low concentration. We covered active transport, which requires energy, requires ATP, and that's because you're moving things from low to high concentration. So here are some lecture questions that I'd like you to understand after this. So I hope that after this, you're able to understand the structure and chemistry of plasma membranes. Again, plasma membranes are phospholipid bilayers, and they're very important in terms of taking up and removing substances, so transport across substances. When we think of the modes of transport across cells, the modes of transport are passive transport and active transport. For passive transport, there's simple diffusion, facilitated diffusion, and osmosis. So simple diffusion is just a passive movement of small or nonpolar molecules from an area of high to low concentration, and they just cross the phospholipid bilayer without any energy, without transport proteins. Examples are oxygen and carbon dioxide. We have facilitated diffusion. Facilitated diffusion is also a passive process, but here you require the help of a membrane protein because the molecules going through are either large, polar, or charged. An example of this is glucose. Glucose is too large and polar to pass through the membrane by simple diffusion, so it moves into cells with the help of a glucose transport protein. And in its example of passive transport because it doesn't require energy. Finally, osmosis is the passive diffusion of water across a membrane, and that's when water is going from high to low concentration. Water moves depending on the amount of solutes. Water will always move where there's more solutes. So if there's more solutes outside of a cell, like hypertonic solution, it will go there. If there's more solutes inside of a cell, water will enter a cell. And if there's equal solute concentration inside and outside of a cell, water will move in and out at an equal rate. Those are all passive transport. Then there's active transport. Active transport requires energy. Active transport requires energy because substances are moved against their concentration gradient from an area of low to high concentration, and it also involves membrane proteins. And then finally, in addition to all of these, eukaryotes also have something called endocytosis and exocytosis, which we just talked about. So I hope that this lecture makes it easier to understand transport across cell membranes.